As a researcher, one of the things that I enjoy the most is to actually be able to share our research, the stuff that we do day in, day out, with the general public. So it is a real privilege for, not only for me, but I'm sure for all my research team to be here with you and try to tell you a little bit of what we do. I'm also excited today because my parents are here. They came all the way from Peru, and this is actually the first time that they're going to see me do a presentation in English. So even though they don't speak English, it should be a good experience for them. Anyhow, today we're going to discuss stem cells. We're going to talk about stem cells and their relationship with cancer. We're not going to talk about embryonic stem cells, which are the ones that you usually see and hear about in the news, which are the ones that are associated with a lot of ethical and political dilemmas. So we're going to try to stay away from that today. What I expect to accomplish in the next 40 minutes or so is to give you basic information so that, tonight, tomorrow, on Sunday, whenever you go have dinner with your family, you can talk a little bit about this so you can be a little more informed about what else are stem cells besides the embryonic stem cells that you keep hearing about in the news. So I really hope to accomplish that. I really want to give you some information, from a scientist's point of view of what a stem cell is and what are the new theories that are coming up and why is it that we're so excited about some of the new discoveries that have been happening not only in my lab but in several other labs throughout the United States and in the world. Okay, I want you to focus on this for a second. So on the left we have an oocyte, a human oocyte surrounded by thousands of sperm trying to fertilize it. Only one of these lucky guys will be able to get in there and form a zygote. It is just remarkable, if you think about it for a second, how is it that a single cell, a big single cell like this, could give rise to a very complex, in this case a super complex individual, in this case such as Mr. Schwarzenegger. So if you think about it, that process from which one simple cell can give rise to a very complex individual made up of myriads of different tissues and organs and systems, it's obviously very complex. So that is called development. A single cell, in this case a zygote, is going to go through a series of divisions, and as development proceeds, that cell is going to commit itself to different fates. For example, at some point in development, the cell is going to say, hey, I'm going to become a blood cell, a liver cell, a brain cell. That commitment, that differentiation happens with time, with development of a human being. And all of that occurs through stem cells. Those decisions, those fate decisions occur through stem cells. So stem cells, as you'll see, are incredibly important, not only for development, but also for regeneration. So once you are an adult like this, your stem cells keep working. In this case, they allow for regeneration of tissues. I'm sure Mr. Schwarzenegger, whenever he hit the gym, needed a whole bunch of muscle cells recruited to form those very nice bulging muscles. So in that situation, the stem cells were working, were being activated, and were being recruited to form new muscle. So if somebody asks you, what is a stem cell? You can give them a very simple answer. You can and sound pretty fancy when you say it. Stem cells are the units of development and regeneration of multicellular organisms. You could do that, right? It's pretty simple. Like I told you, those decisions, those commitment steps that happen through development happen in the stem cells. The stem cells are the ones that make the decision, I'm going to stay as a stem cell, or I'm going to give rise to a differentiated nerve or a lung cell. And also in regeneration, we regenerate our skin, our blood, our gut through stem cells. Okay? So stem cells are the basic units of these two processes. A couple more things about stem cells. Why are stem cells special? And these characteristics apply to every single stem cell, both adult and embryonic. Obviously, stem cells are unspecialized. In biology, we call this primitive. As shown in this diagram here, a stem cell basically doesn't have a very distinct shape like a muscle, liver, or blood cell has. It's immature, if you want to call it that. Okay, that's one key aspect of being a stem cell. Another aspect, which is incredibly important, is that these cells have the ability to produce more stem cells. Okay, and in biology, we call this self-renewal. And we're going to talk about self-renewal more in the latter half of the talk. Okay, so a stem cell can give rise to itself. Okay, so, and if you think about this, then this means that stem cells basically are immortal because they can always give rise to another one of each other. But that's another issue that we could talk about later in the question session. And number 3, obviously, they produce specialized cells. That's another key hallmark of a stem cell. So they are unspecialized, they can produce more stem cells, but at the same time, they can produce differentiated cells. Okay? These cells are non-stem cells. So they usually cannot revert back to the stem cell state. Okay? So please keep in mind these 3 characteristics. Again, stem cells are primitive and differentiated. They can give rise to more stem cells, and whenever they need to, they can give rise to terminally differentiated cells, which are the cells that become the muscles of Dr. Schwarzenegger and the brain cells and everything else in our bodies. Let's look at the development process in a more detailed way. All of us come from a sperm and an egg. And when the sperm fertilizes the egg, we have a zygote. Okay, that zygote is the ultimate stem cell. It's totipotent, meaning that it can give rise to every single tissue in your bodies. Okay? As development and time proceed in human history, that zygote, in this case about 4 or 4.5 days after fertilization, is going to give rise to a structure. It's still an undifferentiated structure, but now this has about 40 to 80 cells. And this structure is called a blastocyst. And from a blastocyst is where scientists can isolate the embryonic stem cells that are on the news. So you can take this blastocyst, which is in fact an early embryo, isolate the cells, put them in a petri dish, and expand them. And a few special cells are going to grow out in the dishes, and those are the famous embryonic stem cells, ESLs. Those cells, in culture, if you give them the right combination of growth factors, they are going to be able to give rise to every single adult mature tissue that we have. Okay, that's a hallmark of an ESL, and that's why people are so excited about them, because they can, in theory, give rise to any major tissue and cell type in our bodies. They are not totipotent as a zygote because they cannot generate extra embryonic tissues, for example, as the placenta. So if a single embryonic stem cell cannot give rise to an entire new mouse or human in theory. Okay? So it's not as potent as a zygote. So just in these 4 days, as we can see, there is a restriction in developmental potential. So those embryonic stem cells, if you compare them to a zygote, relatively, they're still a little differentiated, correct? So as development proceeds, that differentiation occurs more prominently. So a few days after that, then the embryo starts taking a more embryo-like structure. And in this early embryo, the cells are specified into three major categories. We call these germ layers. And one is the endoderm, the mesoderm, and the ectoderm. Okay? And each one of these, in the adult, in the newborn, is going to give rise to exclusive kinds of tissues. Okay, a mesodermal stem cell will commit to blood, muscle, bone, and some other tissues. And so will an endoderm stem cell will do for a liver, gut, and pancreas. Okay? So an ectoderm cell will not be able to give rise to liver. And an endoderm will not be able to give rise to muscle. Okay? So as development proceeds, we're seeing again a restriction in this developmental potential. Once these adult organs are formed, this hierarchy of stem cells does not stop. As we'll see in the next slide, each one of these tissues is also composed of a small hierarchy of cells in which the stem cell is the base of. Okay? So again, to recapitulate, differentiation increases as embryonic development proceeds. Or you can look at it the other way. Developmental potential decreases as development proceeds. Okay? And this is going to be a constant theme throughout human development and regeneration. Now, for example, if we look at one of these tissues, the blood, I told you that each one of these tissues is also hierarchically organized. The hematopoietic system, which is the system that produces the blood cells, the red cells that we have in our bodies, and also the white cells. And you can see a very similar organization as that embryo that I showed you in the previous slide. There's a unique cell. In this case, this is a hematopoietic stem cell. We're also going to call it an HSC. This cell has the highest developmental potential and the less differentiation. And through cell divisions and through, in this case, differentiation, is going to give rise to terminally committed cells that will actually be the functional cells in a tissue. You give rise to your T cells and B cells that are actually going to form your immune system, and also your red cells, for example, that are going to carry the blood and the oxygen in your bodies. So we're going to focus a little bit more on hematopoietic stem cells in the next few slides. But before that, again, just showing differentiation as the stem cell goes down a specific lineage and developmental potential decreases as this happens. Let's review once again what are the major differences between embryonic versus adult stem cells. Embryonic stem cells, of course, are isolated from early embryos, about 4-day-old embryos. Adult stem cells can be isolated from adult human tissues. Embryonic stem cells can be expanded in culture indefinitely. Basically, you can keep the cells in culture for years and years, and people have been able to do that with mice. And the idea is that people will be able to do that with human cells as well. So basically, you would get an unlimited source of cells for repair if you were to have an embryonic stem cell line derived from you. Adult stem cells in general cannot be expanded in culture. I don't think we know at the time if this is just because we haven't found the right conditions to do so, or just because that's an inherent difference in the biology of both cells. This major difference, I'm going to spend a couple of minutes talking about this. Embryonic stem cells can give rise to all cell types, like I showed you, except those extraembryonic tissues like the placenta. Adult stem cells can only give rise to the same tissue from which they come from. For example, a blood stem cell would only give rise to blood. A liver stem cell would only give rise to liver cells. However, this argument was challenged approximately about 6 years ago. People were postulating that adult stem cells were actually, in fact, as potent developmentally as embryonic stem cells. And this became known as the theory of stem cell plasticity, right? Meaning that these adult stem cells had this unexpected capacity to differentiate into tissues outside of their tissue of origin. And this is taken from a paper published by Helen Blau back in 2000. I don't think you can see it very well, but what this suggests is that a tissue-specific stem cell, for example, in this case, the bone marrow and the brain, they reside in each compartment. But whenever they are put on a different microenvironment, in this case, for example, if you put a bone marrow stem cell into a muscle stem cell, the plasticity theory suggested that that bone marrow stem cell could take up the signals from that muscle environment and start acting like a muscle stem cell. And the same in a brain microenvironment, in a heart microenvironment, in a liver niche, what we call it, the microenvironment in which the stem cells grow. So this, of course, had a lot of therapeutic implications, because you could envision that if a bone marrow stem cell will be able to give rise to basically every tissue in your body, then you could envision a whole new array of treatments for all kinds of diseases just by doing a simple bone marrow transplant. And that's important because bone marrow transplants are used widely in the clinic right now. So people started thinking about doing transplantation of bone marrow for muscular dystrophies, which would have been thought of completely crazy just a few years before that. People started doing clinical trials injecting bone marrow stem cells into the heart of infarcted patients. Also, you can think about doing injections for neurodegenerative diseases and all kinds of non-hematopoietic diseases. And I'm going to tell you a couple of experiments that we did in my previous lab whenever I was doing my PhD that studied directly the plasticity of hematopoietic stem cells. So let me tell you a little bit about how we do these experiments. We obviously use the mouse, which is a very, very nice model because of the genetic backgrounds that all the basically genetic flavors of mice that we can have, and just how similar the biology is to that of humans. A typical experiment trying to study the potential of hematopoietic stem cell is to isolate bone marrow stem cells from a mouse that has been engineered to express a blue dye in their cells, okay? And then we transplant that bone marrow into a mouse that doesn't have that expression of that blue dye, basically a non-blue dye. Let's call it that. So we transplant the bone marrow, and then we analyze the liver cells of the recipient mouse months after transplantation. So if in fact hematopoietic stem cells can give rise to liver cells, when we isolate and look in the liver of these mice, we should be able to see blue liver cells, which would indicate that a bone marrow stem cell was able to give rise to hepatocytes, which are the liver cells. And we were actually able to see that. And just to give you a scheme of how we do the experiment, so this is a positive control, what we call, which is a liver section of the donor mouse. As you can see, all the cells light up blue when you stain them with this specific chemical. And in a non-transplanted mouse, you see that there's absolutely no blue. However, in a mouse that was transplanted with hematopoietic cells from a blue mouse, you can see extensive donor contribution, in this case indicated by these blue nodules in here. So in this case, it was true that a bone marrow transplant was able to give rise to liver cells, which was pretty interesting. However, we had some evidence to believe that it wasn't as clear as that. So we didn't think, and I'm not going to get into details of this, but we didn't think that a stem cell from the bone marrow would just go to the liver and start differentiating. We thought, actually, that the cell from the bone marrow would spontaneously fuse with the liver cell, creating this hybrid type of cell. And that is what we would detect on those assays. And in order to test that hypothesis directly, we used a couple of transgenic, genetically engineered mice again. And these mice are very interesting because one of them expresses a protein, a factor, that is not going to be that is going to be inactive unless it sees another factor in this other mouse. So only if there is a fusion between these two cells, the factor from this strain can act on the factor from the other strain and then produce the blue dye that I'll show you in the previous picture. So basically, only if there is fusion, we should see blue cells. So we did the experiment. We transplanted cells from this mouse into this other mouse. Again, if we see blue hepatocytes, blue liver sections, that would indicate that all the liver cells derived from the stem cell came from fusion and not by normal differentiation, which is what people have thought that the stem cells did. And that's what we observed. All of the liver nodules derived from the donor cells were in fact derived from fusion of cells and not from this plasticity, apparent plasticity of the cells. So basically, just reviewing this data, we're able to conclude that hematopoietic stem cells are developmentally committed and are not as embryonic stem cells as people were claiming a few years before. And we were actually able to show that the hematopoietic stem cells were not the ones involved in this process, but actually mature cells that could produce these cell types by cell fusion. And our studies also challenged the rationale behind those clinical trials that were injecting bone marrow cells directly into the hearts of lymphatic patients. So if these cells don't have this plasticity, then why do we study them? Why are these cells important? Why should we spend our time studying these cells? So number one, because they've already been proven to be very useful in the clinic. Bone marrow transplants have saved thousands and thousands of lives. And skin grafting also is helping a lot of people. Also, we study adult stem cells to understand and treat degenerative diseases and also aging. And as I will get into the next couple of slides, to understand and treat cancer. Before that, let's review a little bit our concept again of hematopoietic stem cells. So hematopoietic stem cells in this hierarchy are very rare, okay? Hematopoietic stem cells in a normal bone marrow, they're only about 0.01% of the total cells. So they are very infrequent. Hematopoietic stem cells also do not divide very fast, and that's key, okay, for the tumor stem cell hypothesis, as we will talk about in a minute. And that's counterintuitive, right? Because you would think that the stem cells are the cells that are actually dividing more often to replenish all those tissues. So, for example, if you lose all your B cells and T cells and all your red cells, say if you lose blood, your stem cell is only going to divide very few times. The daughters of the stem cells are the ones that are actually going to expand like crazy, basically. But the stem cells only divide a couple times. And that makes sense from the point of preventing mutations in these very important cells. So these cells are basically quiescent. That's the term that we use to denominate the cells that don't divide very fast. The daughter cells, those are the ones that divide very fast and contribute to regeneration. Okay? So, again, and the final thing is that the stem cells are the only ones that have this capacity of cell renewal, again, of giving rise to more of these stem cells. And again, just reviewing these aspects, stem cells are rare. They are the only ones that have the ability to self-renew, and they divide very infrequently. So, now let's get into cancer. So the big question now is cancer a stem cell disease? And this is not my idea, or the idea of people that started doing these experiments a few years ago. It's a very old idea. And I think it got started by the work of Rudolf Virchow in 1855. He proposed this embryonal rest hypothesis that suggested that cancer cells were similar to embryonic cells in the body. He was able to find some morphological similarities between cancer cells and cells from an early embryo. Now, of course, this hypothesis has been refined, and the hypothesis is that cancer cells basically are blocked in their differentiation, and the stem cells have a lot to do with that, as we'll see. Another important thing to have in mind here is that tumor cells, whenever you put them in culture at very low cell numbers, for example, if you take the leukemic cells of a patient and put them in culture at a very low cell density, probably you see 1 or 2 cells in each petri dish, you would think that if all these cells are carcinogenic, then all of them should form a new tumor. But they don't. A very low percentage of these cells that you see actually form a tumor. Another interesting observation that was done a long time ago was that most tumors, especially solid tumors, are morphologically very heterogeneous. Okay, so not all the cells inside a tumor are the same. Given all this data, still, most of what we know and how we treat cancer right now does not take that into account. People think of cancer right now as basically using this stochastic model. And stochastic means random, basically. And what this model says is that all the cells in a tumor, they all have mutations, and they all have the ability of giving rise to a new tumor if transplanted into a new microenvironment. So as schematized here, if you take one of these orange cells, if you transplant it, you get cancer. The same with a blue or red cell. So it doesn't matter which cell you take, because they're all going to give rise to a tumor. The stem cell hypothesis of cancer calls for something that is opposite. This suggests that there's very few cells in the tumor that we call cancer stem cells or tumor stem cells that are going to be the only ones that, when transplanted, are going to give rise to a tumor. So whereas the majority of the other cells are not carcinogenic in transplantation experiments, as we'll see in a second, only a very low percentage of cells actually give rise to more of these stem cells and give rise to more tumor of the bulk cell. So as you can see, this cancer cell has a capacity to give rise to more cells, which is the self-renewal capacity of a stem cell, and also at the same time give rise to, in this case, not completely differentiated, but tumor cells that are not stem cells. And in the last, I would say, 10 to 10, in the last 5 years, people have been able to do experiments just because new technologies have been available to actually test directly this tumor stem cell hypothesis. For example, the lab of John Dick in Canada has been able to isolate a population of only 0.1% of cells in the entire tumor. Okay, so only 1 in 1,000 cells in the tumor, when transplanted into mice that grow human tumors, can actually give rise to tumors. So that is basically the stem cell for acute myeloid leukemia, and people have been able to reproduce that data. So at least in this case, I think people are certain that there's a very small population of cells that is uniquely tumorigenic. In the last couple of years, people have been able to do similar experiments in solid tissues. For example, the laboratory of Michael Clark at the University of Michigan recently reported that a population of, and these are just the marker of the cell, the cells, the antigens they use to sort the cells, reported that only 2% of all the tumor mass were able to give rise to a transplantable tumor in mice. And in brain cancer, recently, the laboratory of Peter Dix, also in Canada, showed that even though this is not a great enrichment, but it's still significant, that 20% of tumor cells of brain cancer were able to give rise to initiate that tumor. So all the other 80% cells here, and all the other 98, and all the other 99.9% cells in those tumors are not tumorigenic when you transplant them. Again, indicating that within each tumor, there is a very small population of cells that is actually responsible for tumor formation. And here in this diagram, we schematize both normal hematopoietic development and leukemic development. As you can see, the stem cell, again, with the only capacity to self-renewal, and differentiation proceeds as maturation goes. A tumor stem cell, in this case a leukemic stem cell, has a similar organization, similar hierarchy. There's a tumor stem cell that is uniquely within this tumor because it has that capacity to self-renew. This cell will give rise to progenitors and cells that are going to make up the majority of the tumor. In this case, these are called blast cells. Okay? So it seems, understanding this normal hematopoietic stem cell paradigm, we have been able to come up with a map of the hierarchy of this leukemia, this blood cancer. So what are the therapeutic implications of all of this? So most of the drugs and most of the ways, most of the current therapies for people with cancer attack the bulk of the tumor, the majority of the cells that are rapidly dividing. That's what chemotherapy does. It kills rapidly dividing cells. So if the stem cell hypothesis is true, and the cells that are rapidly dividing within a tumor are the bulk tumor cells, then chemotherapy will kill all the bulk tumor cells, as shown here in red, but the stem cells will be left intact, so that with time, those stem cells will be able to give rise and produce more tumor cells. And that is what happens when people get chemotherapy. They get better for a few months, for a few years, then the cancer comes back. So we believe this is the root of the problem. We are not targeting the stem cells, the only cells within a tumor that have the capacity to produce more stem cells and more tumor cells. So what we want to do, and a lot of people are joining in this approach, is trying to target the stem cells within a tumor. And also, of course, give conventional therapy to get rid of not only the bulk tumor cells, but also the stem cells. Without stem cells, basically the tumor would degenerate and would go into remission. So that is the ideal thing. And that is something that we are really working hard on trying to do. And again, if this concept is true, which I think it is because there's a lot of experimental data that supports it already, this will basically change the way we treat cancer. So the big question is, how do we target cancer stem cells? The answer to that is we obviously need to know how the cell renewal is regulated. And interestingly, it's been shown that the cancer stem cells basically hijack all these genes that are important for cell renewal in normal stem cells. So they use that same molecular program. So we need to identify what are the genes that tell stem cells to be stem cells, what are the genes involved in cell renewal. And that is a major ongoing effort in my lab. And just to give you an idea what are the kinds of tools and tricks that we use, these are some of them. We use microarrays, for example. This tool allows us to get a snapshot of all the genes that are expressed within stem cells. This other technique, chip on chips, this allows us to see what are the targets, what genes are activated by some important transcriptional regulators, some genes that are controlling other genes. We can use RNA interference, which is a way of interfering with gene expression so that you can answer the question, what happens when a gene is missing? For example, what happens when in a leukemia we diminish the expression of one gene? And we can do the opposite. We could give more of that gene to see what happens, say, in a leukemia. If we think we have a self-renewal gene, what happens if we give more of that gene? Obviously, the leukemia should become more aggressive. So we have all those tools. And we also can do this in vivo, in mice. So we clearly have nice tools. We have a good hypothesis. So I think in the next few years, we're going to see a lot of nice work coming up from several places that are going to try to identify what are the major factors regulating normal and tumor stem cell self-renewal. And I'll just tell you in the last 5 minutes a small story of one of the genes that we think is involved in normal stem cell self-renewal so far. And the next step is to test leukemic stem cell renewal. So one of the things, like I said, we can do is see which genes are expressed in these stem cells. So this experiment that we did in my previous lab at Baylor College of Medicine, we looked basically at the expression of all genes in the human genome in all the hematopoietic hierarchy. So we took stem cells, we took differentiated T cells, B cells, red cells, all of the cells in the blood lineage. And we selected specifically the cells that were expressed in that stem cell primitive compartment. Therefore, these cells, since hematopoietic stem cells are the only ones that can self-renew, these genes obviously have a higher likelihood of being involved in this self-renewal of cells. And one of these genes is this gene that some other people identified a few years ago. It's called MDS1. And I'm not going to tell you a lot about the gene, but one thing I will tell you is that this gene is a transcriptional regulator. And what that means is that this gene can actually regulate other genes. So it doesn't have an enzymatic function, but it can regulate other genes. So it can tell which genes to be active and which genes to be inactive. And here what I'm showing you is that the expression of this gene is very restricted in the bone marrow. It's very restricted to the cells that are very primitive, to the compartment that has the stem cells. And this is what this is shown here. So we're very excited whenever we looked at this, because we thought, hey, this gene is actually in fact expressed in the primitive cells, and this is in a live mouse. The next thing to do is, again, interfere with the expression of this gene. So we knock down the expression of this gene to answer the question, what happens with the stem cells when you don't have this gene? And in order to do this, we do something similar to what humans do in bone marrow transplants. So you basically take bone marrow cells of your mouse, of a normal mouse, and normal bone marrow cells of a mouse that doesn't have the gene. and then just compare them head to head. If our hypothesis is true that this gene is involved in the cell renewal, in the maintenance of the stem cell state, then the mutant cells would not be able to compete as efficiently as the normal cells. And that is exactly what we observed. And this is just a representative example of some mice that were transplanted with cells. As you can see, wild-type cells are shown in this. Each line represents an individual animal. The percent reconstitution basically means the efficiency of those stem cells are giving rise to blood cells in this assay. So wild-type cells contribute normally, as you can see, and their contribution is steady over time, like a normal true stem cell would be. However, these mutant cells, when transplanted, first of all, at the beginning, they already show a decreased contribution. With time, however, and this is the key observation, this contribution completely diminishes, indicating that the cell renewal of these cells does not happen, so that basically these cells exhaust. And there is a key, that is a key indicator of a problem in the cell renewal of the stem cells of these cells. Another piece of data that is important is if we look in the bone marrow of these mice transplanted with the mutant cells to see if we get mutant cells regenerated, and we don't. So the stem cells are in this quadrant right here, in this square. In cells transplanted with wild-type cells, you see a good population of stem cells. However, in the cells transplanted with the mutant cells, there's none. There's no stem cells that have been regenerated. Again, indicating that a lack in the capacity of the cells to give rise to more stem cells, meaning a cell renewal defect. We can also study these cells in culture. So we can put the cells in culture and just see what happens with wild-type cells or with cells that are deficient in MDS. And what you can see here is that on top of this line right here, it's differentiated cells, and the bottom, undifferentiated cells. So on a normal, and just focus on the right side of each panel, forget about the left side. On the normal cells, we have about 57% of cells that have differentiated in culture. However, in the mutant mice, in the mutant cells that don't have this gene, about 90% of the cells have differentiated, indicating that cells that don't have this gene, basically they all differentiate. They cannot give rise to more stem cells, basically. Okay? Another piece of evidence that suggests that this gene is very important in maintaining that cell renewal and maintaining that stem cell state. Obviously, there are big questions that we're currently working on. The big question is MDS1, this gene that I told you about, is that also required for the cell renewal of leukemic stem cells? And we're currently working on that. Obviously, what other genes are regulated by MDS? Should we, maybe we can identify new regulators of cell renewal that could be targeted by drugs in the future. And with that, I would just like to introduce the members of my laboratory. Sandra Stelling is a postdoctoral fellow that just joined my lab a few weeks ago from Germany. She comes from one of the most prestigious institutions in Germany, and she's doing very exciting work trying to determine the mechanism, which is the way how MDS1 regulates the cell renewal of these stem cells. And then at the same time, she's trying to identify novel genes that are involved in the cell renewal of these stem cells, and therefore in leukemic stem cells. Jonathan is a technician in my lab. He just graduated from Harvard in May, and he's doing some very, very cool work in which he's trying to study the fate of individual cancer stem cells in vivo in a mouse to see what happens with each one of those cancer stem cells that you transplant. Those studies are going to be great. He's also trying to take a look in more detail on the nature of these stem cells. How biologically, how different are they than normal stem cells, and how similar are they? So if you have a chance to talk to him, he has some very, very good stuff that hopefully gets resolved in the next year or so. Two people that I didn't have a chance to take pictures is Luis Alvarez, visiting scientist in my lab. He's working on trying to expand hematopoietic stem cells. Like I told you, adult stem cells right now cannot be expanded significantly in vitro because they differentiate. So if you put a very good, strong stem cell in culture, in a few days, you'll have a mature cell that is not good. So he's trying to use some of the genes that we've discovered and some of the genes that other people have discovered. He's trying to interfere with expression of those genes so that we basically are able to expand these cells in vitro for a period of time and then revert those cells back to the normal state before transplanting them. Andrew Green is an undergraduate from MIT working in my lab, and he's been involved in some of the experiments that I told you with the transplants with MDS1 and the whole story. Andre's been great. He was the first member of my lab, and hopefully I can keep him for a long time. He's just a freshman. I mean, he's a sophomore now. But if you have a chance to talk to him, he's really great. He's really passionate about science. I would just like to finish acknowledging the people that have been able to, have allowed me to do this. First of all, Margaret Goodell, which was my advisor at Baylor College of Medicine. We still have very strong collaborations with her, and she just does great science. Archie Perkins at Yale, we have a very good collaboration with him studying MDS1 and another partner gene with MDS1 called EV1. The Jenish lab at the Whitehead Institute, these two people have been fundamental in doing some of the work that I showed you, and the Bromelcan lab also at the Whitehead Institute. With that, hopefully you guys enjoy this, and I'll be really happy, and they'll be really happy to take some questions.
So we'll take some questions from this audience here and then a few questions from the Worcester audience as well. Does anyone have any questions?
Why is there a need to remove the non-stem cell cancer cells? You said that you want to get rid of all of it.
Right.
Okay.
So the question is, in the diagram that I showed you, I suggested that we should attack both the tumor stem cells and the non-tumor stem cells. In theory, we should be able to just get rid of that tumor by just tackling the tumor stem cells. Because basically we'll have a tumor mass that shouldn't be able to produce more tumor cells. But again, if we do a combined therapy, we should be able to get rid of both faster, basically. But in theory, it should work like that, like you suggest. And the question is, if the non-stem cells in a tumor are rapidly dividing, why is it that they cannot form a tumor themselves? That's a very good question. And we could answer that just by looking at the normal biology of stem cells. For example, the hematopoietic system. Like I told you, in the hematopoietic system, and let me go back to the slide, the stem cells divide very rarely. The cells that divide a lot are these, basically what we call progenitors. Okay, these cells divide very fast. However, they only have a very limited lifespan. So for example, if you transplant these progenitors, you're not going to get long-term reconstitution, long-term engraftment in mice or even in humans. So even though they divide very fast, they basically exhaust after a while. And we think the same thing is happening with tumor cells. So even though they have a burst of proliferation, basically, they can proliferate a lot, they cannot sustain that for long periods of time. Now, regarding your second question, it's another great question. Are there genes that seem to be common for all the stem cells, regardless of the tissue of origin? A lot of people are working on that question. My previous lab did some of that work, and it actually seems that first of all, nobody has been able to find such a gene as of yet. There's a couple of genes that have been found to be expressed in several types of stem cells, but I don't think there's one gene that people would say this is a stem cell gene found and required for all stem cells. People are still doing those experiments, but again, sometimes they're very hard to do because stem cells are very rare. So you need to have a very pure population if you actually want to look at all the genes that are expressed exclusively in the stem cell. And we only have that very pure population for the hematopoietic stem cell system. So for example, in case of skin stem cells, I think people have ways to purify them, but that population is still very crude, very impure. So out of that population, a very low percentage of cells would actually be a true stem cell. So basically, if you look at the genes that are expressed in that population, you'll be looking at some genes that are not stem cell specific.
Can we take one question from Worcester?
The question is if I could just comment a little bit more on the plasticity of adult stem cells. So basically the fact that, say, a hematopoietic stem cell could give rise to non-hematopoietic tissues, or that a liver stem cell could give rise to non-liver cells. I'll show you some evidence that this doesn't happen. But this is a very controversial issue in the stem cell field. There are people that have nice experimental evidence that shows, for example, that hematopoietic stem cells can give rise to liver cells by a mechanism that is not cell fusion, but a mechanism that is just cell differentiation, just like an embryonic stem cell will give rise to a liver cell. People are, I guess the jury's not out, but in most cases, even if this happens, it seems to be a very infrequent event, an event that occurs very randomly. And I personally don't think it has a very big physiological meaning. I think the lesson to be learned from this is that maybe if these stem cells can in fact be reprogrammed, which I think is the case, maybe we should learn how that happens, and we should try to manipulate that as opposed to let the body do that work. We should be able to manipulate this transdifferentiation so that it occurs in a more efficient manner.
Quick question about the profiles of markers that you were pointing out that are associated with the different tumor stem cell types. How much or how often are those found across clinical samples? So do all brain tumor stem cells from various patients all express CD133, et cetera, et cetera? Exactly.
That's a very good question as well. So tumors are very heterogeneous. So even leukemias, an acute myeloid leukemia is different than acute lymphoblastic leukemia. So each one of these cases that I showed you is specific for a specific type of tumor. Okay? In the case of the acute myeloid leukemia example that I showed you, which is also called AML, for this AML, people have been able to reproduce that data. And that's CD34 positive, CD38 negative, and I think there's another marker, I believe CD20, I'm not sure. People have been able to do these experiments reproducibly, get samples from several different individuals and do these experiments. And even AML has several stages, and I actually think people have been able to do that with several of these stages. But it doesn't seem that these markers are conserved if you go, for example, to acute lymphoblastic leukemia. So I think the way people are going to try to sort this out is by looking specifically at a very, very specific type of cancer.
Are the cancerous stem cells in a tumor going to be more resistant to chemotherapy than the rest of the cells in the population in the tumor?
That's a great question. So I don't know if I could go back to this. So actually, since we're here, so hematopoietic stem cells, like I said, they don't divide very fast. And chemotherapy agents, they kill dividing cells. So in fact, if we kill dividing cells, we are not killing the tumor stem cells, because these cells divide very rarely, so they don't even get affected. So we're killing the whole mass of the tumor, but not the tumor stem cells. So we need to basically come up with different agents, because chemotherapeutic agents won't work. Moreover, it seems that stem cells and some of these tumor stem cells express genes that allow these cells to be resistant to to any chemotherapy agent. They express these proteins that allow the cell to extrude, to actively pump out the drugs that you're trying to give them. So again, it's going to become very important to try to determine the transcriptional profile, so what genes are active in these cells, so we can specifically target those. The question is there any evidence that the tumor stem cell, in this case the leukemic stem cell, is derived from this hematopoietic stem cell that has mutated, or a more differentiated cell that basically with the mutations of cancer has turned into a stem cell, but in this case a cancer stem cell? That is currently under investigation by several labs. But if you just think about it, it would seem much more likely that the mutations originated and accumulated in a long-lived population, like a hematopoietic stem cell. Because, for example, if you have a mutation in this mature macrophage, macrophages only have 24 to 48 hours of lifespan. So basically, you need more than one hit usually for cancer to occur, more than one mutation. So if this happens here, the chances are very, very unlikely that this macrophage will get another mutation that would allow it to survive and produce a full-blown leukemia. But if a mutation arises in a hematopoietic stem cell that is there for the lifetime of an individual, then during that lifetime, maybe a second and a third mutation can arise. And then the stem cell will probably be derived from a stem cell. There's some evidence that suggests that that is true in fact, but also these cells, the progenitors that have this high proliferative rate, although a limited lifespan, can also be the target of some mutations in some leukemias.
You mentioned that stem cells could be kept much longer periods of time, almost indefinitely. I'm wondering what characteristic of a stem cells allows them to be kept for such a long period of time?
That is a very good question. So again, I don't know if the inability of adult stem cells to stop growing after a while, differentiating basically in culture, is due to the fact that we just haven't found the right ingredients, right, the right cooking mixture that they need in order to be happy in culture, or is just something inherent of the cells. Embryonic stem cells, in fact, can proliferate for years and years and years. So people, for example, have been growing lines of mouse embryonic stem cells since the 80s, and those lines, they can still contribute to a mouse if you inject them into an early mouse embryo. Why is that? Number one, embryonic stem cells express telomerase, which is an enzyme required for keeping your chromosomes, your DNAs, together, basically, to keep them happy after each cell division. So embryonic stem cells and some adult stem cells express telomerase. Also, I believe a key aspect is to maintain the cells undifferentiated. And in embryonic stem cells, people have identified a molecule called leukemia inhibitory factor, at least that works in mouse ESLs, that keeps the cells undifferentiated. So it blocks that differentiation process. So because if it goes under that pathway of differentiation, then the cells, since they become differentiated, are going to stop proliferating. So I think it's a mixture of both the factors that are necessary for a cell and the intrinsic genes that they are expressed.
The MSD, is that correct?
MDS.
MDS, does that regulate through transcription factor, or what does it regulate through?
Yes, MDS1 is a transcription factor. So a transcription factor, for people that are not familiar with, is a gene that codes for a protein that binds DNA sequences of other genes and either tells them to be on or to be off. So it's basically what is called a master regulator. And in this MDS1 in particular has some nice sequences that tell us that possibly it's a major regulator of DNA conformation. And that also allows some gene expression and gene shutdown. So we're currently doing experiments trying to determine exactly how is it that MDS is doing this transcriptional regulation.
Embryonic stem cells can form any cell. Is that correct?
The question is, can embryonic stem cells form any cell? The answer is no.
No. Okay.
They cannot give rise to extraembryonic tissues. And let's talk about the mouse for a second, okay, since we have years and years of experience with that. If you inject embryonic stem cells into an early embryo, all of the adult tissues of that mouse will be derived from that embryonic stem cell. However, a single embryonic stem cell cannot give rise to an entire mouse by itself, because it cannot give rise, for example, to the placenta, which is necessary for embryo development. It can give rise to all embryonic tissues, basically, so meaning all tissues in the adult.
So the embryonic stem cell can create all cells, and there are external stimuluses that cause it to create those cells.
Exactly.
But the zygote is the only cell that can actually differentiate into an entire organism.
Exactly.
Okay, thank you.
The question is, what is the reason why some tumors are heterogeneous in morphology, like I mentioned to you? This, I think, happens to be in most cases, and the literature, there's from almost a century, just analyzing the tissues of people with tumors. And if you just look at basically the tumor bulk, where all your tumor cells are, not every single cell has the exact same shape, basically. And that indicates not only that they have a different morphology, but also they might have a functional difference. Why that is, we think this explains it. There's a few cells that are primitive, the tumor stem cells, and the other cells that, even though they are partially blocked in the differentiation program, they might resemble a little bit more the normal differentiated cells. Let's remember, cancer is a problem in either differentiation, so the cells cannot differentiate. So the cells are going to be blocked at some sort of developmental stage, but the fact is that there are different looking cells within each tumor. And I think that is true for most of the tissues that I'm aware of. Do we know how do cancer stem cells respond to other cancer therapies, such as radiation or anti-angiogenesis drugs? That is a very good question. I would say there's very limited data on that, because this whole new concept of cancer stem cells, it's just at the most 2 years old. So I don't think people have actually done those experiments. So, but still, it seems that, for example, after irradiation, you still get tumor regression. You never get like complete tumor ablation. Angiogenesis is anti-angiogenesis drugs are still a very experimental approach. So I think people will be looking at that right now, and they are doing that right now. And maybe anti-angiogenesis drug will actually be what will be most effective to kill these cells, or maybe something else completely novel.
Okay. So how would you describe the process of metastasis using this theory? Is it like the tumor stem cells are actually what travel through the lymph or the blood?
Right. Very nice question. We get some great questions. I don't know if somebody gave you a piece of paper, but these are just better than in a scientific symposium. Seriously. Wow. The question is, how do we explain metastasis using this tumor stem cell hypothesis? We've been thinking about that a lot, and actually Jonathan is trying to make these mice that will track individually unique cells in a tumor, so that we could see which cell within a tumor is actually the one that's going to give rise to the metastasis. We do think it's probably a tumor stem cell that migrates and basically lodges into a different site, but that nobody has ever shown that, and that definitely awaits experimental verification. The question is, are all of those tumor cells, for example, in acute myeloid leukemia, derived from a single cell, which we call clonal? And I think there's been years of work that have suggested that all tumors are clonal. So basically, the mutation happens in one cell, and then tumor genesis starts from that single cell. And that, I think that is still basically the paradigm. The way we accommodate this hypothesis to that is that mutation, say in case, for example, of CML, you get the BCR-ABL translocation, that happens in a stem cell. And then that stem cell acquires more mutations, and then that gives rise to a tumor. But that initial mutation, and therefore all the tumor cells come from one clone, from an individually mutated cell.